All right, thanks everyone joining us in person and on Zoom today. We're really pleased. We got very lucky. So one of our newest Steve colleagues, Ari Iyer, had his colleague, Charlie Roscoe, visiting and asked us, could we please host a talk, because Charlie has a lot of interesting data to share. So we're really happy that you're able to come to visit us today. So Charlie is currently wrapping up a postdoctoral fellowship in the Department of Epidemiology at the Harvard T.H. Chan School of Public Health. She did undergraduate work in biology in the UK and a master's in ecosystems and biodiversity in France and in Belgium, after which she returned to the UK to Imperial College London for her Ph.D. in environmental epidemiology. She uses spatial techniques to disentangle environmental exposures from socioeconomic factors and is particularly interested in cardiovascular disease epidemiology and prevention, as well as now doing work in cancer epidemiology. She's published extensively on how the environment impacts health, including really interesting work on the benefits of various types of green space and walkability using large cohort studies. Dr. Roscoe is soon to join the faculty at Oregon Health Sciences University, OHSU. And we're really pleased that you could stop by Rutgers today to talk with us. I'll turn it over to you. Let's see. Are you here? We have a click. So click a clicker if you want one, but otherwise, okay.
Thank you for that lovely introduction, Emily. Can everyone hear me okay? I should project a little bit. Okay, yeah, I'm very excited to be here. This is my first time at Rutgers and in New Brunswick. And yeah, let's get started. So today I'll be talking about chronic disease in the context of the environmental exposome. If any of the words that I talk about are new to you, you will know what all of them mean by the end of the session. As Emily said, I'm finishing up at Dana-Farber right now, some projects with Harry, and we'll continue to work into the future when I'm at OHSU PSU, which is a new school of public health in Portland. That's OHSU is the kind of very clinical center, TSU is the Portland State University, and they have joined forces to start a new school. Okay, so some content. Who am I? Emily's just done a great job of this, introducing me, so I can probably skip these slides. The impact of the neighborhood environment on chronic disease. I've mainly focused on cardiovascular disease and recently started looking at prostate cancer outcomes. I also have done a tiny bit of chronic respiratory disease stuff in my PhD. I'm going to talk about Earth observation for epidemiology. If you don't know what it is, you will by the end. I'll talk about modeling environmental interventions to reduce prostate cancer outcome disparities, and go over some future research on the exposome that I'd like to develop when I start as a professor. Okay, so we've already had this summary basically, but I was born and raised in Oldham, which is east of Greater Manchester in the UK. You can probably detect an accent. I did my master's in Paris and also Belgium. I was a research assistant at the National University of Singapore. I worked with a landscape architect there and got very interested in biodiversity and green space. Mainly focusing with her on people's comfort level in different levels of rewilding, and that really triggered me to get very interested in green space and health. Then I saw an advert for a PhD in UK Biobank that was in green space and health, so I was like, okay, I'll apply for this one PhD. Somehow managed to get it, great, moved back home, and now I'm a postdoctoral fellow. So I'm telling you this bio because I want you to know who I am, but also I want to give some context as to why I'm motivated to look at disparities. So as I said, I'm from Oldham, which is one of the most deprived towns in the UK, always top of the league for deprivation, also has a really good football team, so sometimes it's top of the league for football. It's just outside of Manchester, so some of those players end up in Manchester and playing on big teams. So we measure deprivation in the UK by 7 domains that make up the index of multiple deprivation. Those are income, employment, education, health, crime, barriers to housing and services, and living environment. And now I got interested in health and health disparities from growing up in that context and just seeing how different in the UK, there are racial disparities. There's also very strong class disparities. It's very hard to move from being a lower class person to a middle class or upper class person, and they remain very much intact. Although we don't talk about them, or we do as though it's a historical thing, but it's not historical. That building on the left there in the picture is a cotton mill. So many of the mill workers in the town that I'm from, including my great grandma, the industry collapsed essentially and was moved to cheaper places to produce, usually in South Asia. And it left a lot of people kind of jobless. You can think about Detroit as a kind of similar thing that happened with the industry there. Anyway, so that's my background of how I got into environmental health and disparities. And I also wanted to show you this because it gives a nice introduction to something that I'm starting to get more interested in, which is life course epidemiology. So if you took me as an individual and you measured right now my environmental exposures, my social exposures, my income, I'm not doing that bad. I'm in America. I live in Boston now. I actually live in Brookline, which is just right on the side of Boston. It's pretty nice. I have a park nearby to where I live. It's not too polluted. I would not look anything like I looked as a child, but whether those determinants of health, those social determinants of health that I had in my childhood impact me now is unknown. And often in a cohort, what would happen is I wouldn't even be eligible for a lot of cohorts if they wanted to measure adult health. They would want, people who are more likely to develop chronic disease, who are in their 50s or 60s. And then by that point, I probably live in Oregon, and I won't look anything like where I was from. So part of this is kind of thinking about life course epidemiology, residential histories, and this kind of work that we're trying to kind of link up developmental origins, so early life exposures and kind of later life onset of chronic disease. So that leads me very nicely to the bit of the exposome that I would say I have the most experience on working with. So life course exposure assessment, if we try to think about geospatial exposure assessment, which is where most of my expertise have been. So you have a place for an individual, and often that is somebody's residential address. You may have one, you may have residential addresses across their life course, but in that place that you have, also people are not static. So maybe we want to add to that and have kind of mobile technologies letting us know really people's activity space and where they go. But say you just have this one point in space and it's their residential address, and you have all of their exposures, their light pollution, their noise, their air pollution, the green space, the population density, whatever factors you might want, neighborhood socioeconomic status. Even if that person stood in one spot in their house and never moved ever, they just stood there for their whole life, which is not very realistic. You still would see change in many of these exposures over time. So definitely, you might recognize these climate stripes on the right. The climate stripes show the increase in temperature over time. So those are annual averages, and it's getting hotter, as we all know. So even if you did stand in the same place over time, you'd still want to update your exposure assessments temporally, because you have shifts in these exposures. And you can also leverage that some exposures do change more than others due to policies or due to whatever. It can be natural, it can be unnatural, but things such as parks, for instance, are probably pretty set in space and don't change that much over time unless they get demolished and built on, but tend to stay intact, whereas maybe the air pollution on the road next door really depends on the policies and EVs versus bikes versus blah, blah, blah. So you can really kind of try and pull out those different exposures and try and tease apart what actually is causing health problems, what is benefiting health if it's green space and okay, so sorry, did that work? So I'm going to give you a quick highlight reel of my studies. Hopefully I'm not going to go over any of them too fast, but I'll answer in the questions if anyone has any questions. So I'm going to talk about just generally the impact of the neighborhood environment on chronic diseases. And when I did my PhD on green space and health, I don't remember which year I started, but it was at the time where there was some kind of early green space literature, and the mechanisms were kind of hypothesized, not dug into too much. So I started off and I thought, okay, green space is linked to these chronic disease outcomes. A lot of them were ecological studies at that point. So I got the opportunity to work on the UK Biobank, which I'll introduce in a second, but essentially that's a cohort of 500,000 adult individuals in the UK. They were all over 49. And they are followed over time and gave questionnaire data, gave biomarkers, came in for an interview, and they have all of these massive amounts of questionnaire data on their lifestyle. And I got to thinking, okay, these mechanisms, which ones can I best measure in UK Biobank at this point? And I thought physical activity and environmental. So if we have our green space on the right over here and linked to chronic disease, there were hypothesized mechanisms of increased physical activity, cognitive restoration, stress reduction, attention restoration theory type stuff in the psychological benefits. For social network, you go for picnics, you might go on a walk with a friend, you might bump into your neighbor and talk to people. So that can be good for health. Biodiversity at that point was very weakly evidenced, but it's getting stronger now that there is some epigenetic effect depending on the microbiota that you're exposed to. Climate mitigation, obviously trees can help with shade, temperature reduction, SUDs are sustainable urban drainage. So when we have flooding, they take up a lot of the water and can help with offsetting the health problems associated with that. And then environmental is kind of the buffering of things that we know are already, we already have a lot of evidence that air pollution is bad for our health, noise too, and greenness can, depending on how it's arranged, can buffer environmental exposures. I will just say it can make it worse, which a lot of people are saying that trees are going to solve everything, but if you put trees over a road in a tunnel, it will make the air pollution in that tunnel much worse. So there needs to be a lot more nuance as to how we talk about greenness and air pollution, because I think sometimes it's a bit of it's going to save the world, and it could if we do it right. Anyway, so in the interest of time, I'm not going to talk about the environmental section of this. I'm going to talk about physical activity. And then I was thinking of the pathway-specific markers in UK Biobank, which would be the questionnaire data, so self-reported data. They also have accelerometry data on about 100,000 participants. And then they also have some internal biomarkers, kind of C-reactive protein, inflammation markers from blood biochemistry, and some fitness stuff. So how we would usually do this in the UK Biobank is we have residential addresses of those participants, individual level residential addresses. We draw a circle around their home at a buffer that is, a priori we've said, okay, I think it's physical activity, and maybe within 1 kilometer, people are using the green space. So basically we make a good guess and measure the greenness within that buffer. So I started doing this and started thinking about it, and we had this nice data set that was split into trees and ground cover, which is like greenery that's lower than trees. And then I was like, I was reading the literature and I was like, it's very mixed on green space and physical activity. Some of it's showing that it does increase physical activity, some of it's showing that it doesn't. And I got to thinking, why? And basically we know that walkability is very much linked with people's physical activity. So if we look at greenness, which is often inversely correlated in space with walkability, because walkability is how many buildings there are, how many shops, how many people, how many road intersections, which are all not green spaces. Apart from street trees, but get to that. And yeah, so I started trying to think about this a little bit more in depth as I was doing the exposure assessment, and I didn't have access to any of the physical activity data at this point. It was a decoupled exposure assessment and then get the health data later. So I was thinking about this and I was like, okay, I'm going to integrate a walkability exposure. And in the end, I ended up producing this walkability exposure that was based on those 3 things that I just said. So business density, population density, road intersection density. So a very simple, straightforward walkability exposure that's been well validated in the London older adult context, at least. And then I thought, okay, if I add in trees and ground cover, so greenness into the buffer of measure walkability, then you can see the kind of incremental additional effect of adding green space into that. So this is a lovely little map of one of those points, which actually isn't a UK Biobank address also. And what we found is that participants were more likely to achieve their national recommendations of moderate vigorous physical activity and walking, which combined is 150 minutes per week in the UK. 36% more likely to achieve that. They also, using their accelerometry data, had an additional 2.41 additional minutes per day of moderate vigorous physical activity, which might not sound like a lot, but in older adults, that's a lot. So that was comparing the fifth quintile with the first. So the highest green walkability with the lowest. And what we saw is that most of the effect was walkability, but we did see a supplemental effect of the greenness, especially street trees, because we could pull apart the trees and the ground cover. And yeah, anyway, I'll leave that one there. And as Emily said, I have been very lucky to work on UK Biobank and Green Space, and the UK has some really nice data sets. So it has Ordnance Survey Master Map, which is the equivalent of the kind of government USGS probably, has this really nice data set where they have the green space and they have attributed the type of green space. So I think this is the first I've ever heard of this sort of data set at a national scale. So I looked at that, and the reason we looked at this essentially is because everybody, when they saw this NDVI green space research, greenness, vegetation cover causes, helps prevent this many deaths, etc. Everyone's mind is like, oh, parks, that's what you think. You think parks, greenness. But the majority of greenness in cities in England and many places are private residential gardens. So a lot of the green space in the kind of buffer sizes, the circular buffers that I was showing at the start, that kind of distance around homes, a lot of what the greenness is there is private residential gardens. So essentially we added this into UK Biobank. It's there for any researchers to use now on over 300,000 or so urban participants. And we saw that indeed actually I have a graph. We saw that indeed, all green space is good, but private residential gardens, even though the confidence intervals overlap, does appear, the trend appears that private residential gardens are kind of driving this a little bit more than other green space types. So just to orient you, you have the hazard ratio on the bottom and the dotted line at the 1. And then these should be italicized, sorry, but all-cause NES, non-injury, which NES cardiovascular disease, which NES IHD and cerebral stroke. And then respiratory, also NES chronic respiratory, which NES COPD, and chronic and respiratory also NES infectious. So sorry, they should be a little bit different. But we didn't expect to see anything for infectious, which we didn't really. It was pre-COVID. So most mortality from respiratory at that point was from pneumonia in the hospital. So we didn't expect residential greenness to really have a big impact there. What we did see is some pretty strong effects, especially for COPD. And you can see that the private residential gardens, the dark blue line, is a little stronger than the other green space, and they are mutually adjusted for each other. So others, including parks and kind of public green spaces. So all of it's on the protective side, but definitely just reframing people's thinking as to what that means. Obviously, we don't want to encourage sprawl, so we didn't say everyone should have a massive private residential garden in UK cities. But what we said is that there may be some benefit to gardening, or if that's horticultural therapy, having an allotment or a community garden that you can participate and have some sort of ownership feeling. Potentially, that is helping with health as well. And I'm going to just flip to what I'm working on now in green space. This is all unfunded, but hopefully funded soon. So I've just been invited to join the Green Me or Green ME project, which is very big it's a Horizon Europe project. So it's lots of different universities in Europe looking at green care, which is like horticultural therapy or things like that, effect on mental health. And they did have a US partner who has dropped out, and then they asked me to join. So good for me. Sorry to the other partner, I don't know what happened. But probably I'll think about like the VA and horticultural therapies. But if anyone has any ideas, it's very much in the early stage. It's just been awarded. So happy to hear if there's anything in New York or in New Jersey that you can think of as examples that I could bring up to them as kind of, they are saying that the US is the leader in this kind of thing because we have programs in horticultural therapy. We have very established ways of running these programs which don't exist yet in Europe. So they are looking to us to inform how they should tell the European Union that this should be you should be able to prescribe this, you should be able to kind of offer this as a healthcare service. Kind of new area for me because I haven't done mental health before. And then I'm also looking at biodiversity exposure. So I got interested in that biodiversity mechanism. And finally, there is a data set that's less than 1 kilometer resolution across the contiguous US. So I've joined that to the Nurses' Health Study, which I'll get to in a second talking about, and I'll see if there's anything there with the microbiome samples. You can imagine it may impact people's microbiome, especially if they've taken antibiotics or something that, they become very susceptible to their environment, and cardiovascular disease outcomes, partially because I've just already looked at that, but also I'd like to look at cancer as well. So that brings me nicely on to exposure assessment in the Harvard cohort. So I've been at Harvard for more than 3 years now and done a lot of spatial joins. And one project that a colleague was working on was on temperature. And I ended up working on the exposure join for that. So temperature, obviously, as we know, is changing over time. The temporal element, as I showed earlier, is very important. So as much as annual averages might be useful, we might want monthly or something like that. And just to say what that actually looks like in the Harvard cohorts. So the Nurses' Health Study, I don't know if you're familiar with Nurses' Health Study, Nurses' Health Study 2. There's also Nurses' Health Study 3, but I haven't included that on this slide. And HPFS is the Health Professionals Follow-Up Study that Harry's done a lot of work in. And altogether, there are many participants, and we have residential addresses for those participants. They're followed on questionnaires, and every 2 years we get an updated address if they've moved address. And we collect all of our information during that process, but we geocode that residential address so we know if they're moving around. And that's been collected since the 1980s for nurses and nurses too. And altogether, we have more than 1 million points just for those 3 cohorts, not including NHS3, which is a lot of points in the first place. And then if you want to also add monthly temperature, and you want to do that over approximately a 21-year span, depending on the follow-up of the participant, and you have 7 variables, it turns into this kind of big chunk of work. So a lot of spatial joins, as you can see at the bottom here. And this is my colleague, Greta Wilt, who helped with that process, and we built on some of Harry's process for linking. He'd already linked greenness to the cohorts with another colleague, Isabel, and so that's what it looks like. And we don't only join temperature, so we've also joined greenness and kind of social indices, and got the social vulnerability index here, walkability index. That's a bit blocky because it's at the census level, and noise. So another project of mine while I've been at Harvard, which has been provisionally accepted finally just the other day to EHP, is a study on noise and CBD. So this is just one example of one of the nurses studies that we've worked on, following nurses from 1988 to 2018, which is the last time we have any geocode information. Actually, we have it, but it's not geocoded yet. So that's a lot of person years follow-up, a lot of CBD events in those nurses. There was around 120,000 nurses at the start of follow-up. And so what we see is we have a noise model. It's by the National Park Service. It's got flaws for sure, but it's contiguous, and it gives an estimate of noise, which has been validated. But it shows that with all of the amazing confounders that we have in the nurses and the longitudinal follow-up, we found that there's a 4% increased risk of CVD, cardiovascular disease, which is basically CHD and stroke in the nurses. That's validated for long-term noise, so annual average noise. And we see very similar for nighttime and daytime noise levels. So that should be out soon. Hopefully will help me get momentum to maybe do some modeling on noise that's a little bit better than what we have already. That's the hope. Okay, so I'm going to switch really quickly to Earth observation for epidemiology. And I love Earth observation, so I'm going to try and keep this as brief as I can and not go over time because I'll get excited. So Earth observation is essentially satellite imagery collected. This is GPS. This is the Landsat that is used to collect kind of images that we get different spectral bands. So all the vegetation indices that you may see published comes from Landsat, usually this satellite on the left. And it's really changed the game in terms of exposure assessment. They go around the world and, depending on what time frame and where they go, you have repeated measures essentially perhaps every 2 weeks or something, which for us is what we love as epidemiologists, because then we have repeated measures of exposure. It's not intrusive for our participants whatsoever. And we can also get their GPS, depending on your study protocol, can be hard to get. But if you have GPS of people moving around, then you have more than just their residential address to join their exposure to. So, as I showed before, the way we measure green space so far has been in these residential buffers, so these circular buffers. And as I said earlier, you kind of guess which one is relevant, which is not an ideal scenario. And many studies can't afford to have GPS or can't get ethics to have GPS on their participants yet. And you don't know exactly where these people are going. So if you've got your standard study design where you have these circular buffers and you're using your satellite data to measure how green it is around the residential address, we wanted to work on a problem that was called the uncertain geographic context problem at the start of my postdoc here at Harvard. And that essentially is, how do you choose? Like, which context is relevant? The wider buffer, is the buffer right around the home relevant? How do you choose? Usually we'll say a priori, oh, I think it's Earth, so we'll do a tight buffer, but really it's just guesswork. It's not, and it opens you up to, problems of fission. If people look at 9 things, maybe they're just going to choose the strongest one and publish that. So we wanted to, I'm not going to go into this, but we wanted to look at how to do this in a standard longitudinal analysis, so using a Cox proportional hazard model. And we came up with some penalties and a way of, I worked with a bunch of statisticians who are at Yale, and essentially if you put in the buffers into this all at once, and this kind of penalty can select which is the appropriate buffer for your data that you're putting in. So it's not a fishing thing. It's not giving you the highest hazard ratio. It's giving you, it works exactly like a Cox proportional hazards model. It just penalizes the buffer size to help you select the buffer. You get a confidence interval on the buffer, which I'll show you is quite wide, but at least you get an estimate of which is the best buffer, the confidence interval, and then you also get your hazard ratio out of that as you would in a standard Cox proportional hazard model. Okay, so in our, I'm not going to go too much into the detail, but it was in nurses' health study again. It was on greenness and it was on depression incidence, because we'd already seen something when we were doing that kind of guesswork buffer thing, that like the smaller one was better, the bigger one not so much, but like we were just guessing essentially and giving a priori mechanisms. So when we did this, what we saw is that the greenness within the 90-meter buffer distance was associated with depression, but we found that that was after that buffer, there was no additional information of adding extra exposure data. So when we added the outer rings, that gave us nothing extra after 90 meters, which happened to be our smallest in this cohort. Now that's specifically for depression incidence in nurses' health study and would need to be replicated over lots of studies, but it might give us some kind of clue of what mechanisms. Maybe it's visual, maybe we need to be able to see it for it to impact depression. Maybe you think it's socioeconomic confounding, which I think is the case a lot of the time with greenness around the house, especially knowing that it's private residential gardens. But whatever it is, it's best, or it's explained already in that first 90-meter buffer, and that's what this method adds. Yeah, so we saw a 3% decrease in depression risk was observed for each 0.1 increase in NDVI, which is a normal measure to measure NDVI. It just means it's more green. And the nice thing is you get your normal standard outcome, but you also get, I told you they were wide, you also get a confidence interval around your buffer. So the buffer selection itself, it's definitely saying that it's on the lower end, kind of. I mean, the buffer's wide. Okay, so that leads me on in this kind of Earth observation, satellite data, using that, and then I said the ideal scenario. So additionally in this grant, we're going to say we do have data on where people are moving, so we don't have to just use this residential address and try and get that as good as we can. We do have data in Nurses Health Study 3 where they wore Fitbits and they had a BWE smartphone app, and we tracked them for 4 weeks, spread throughout the year in different seasons to see what they were doing. The Fitbit measures physical activity and sleep and heart rate, and the smartphone is just basically following them around. It has some little bits of questionnaire that you can add to that. And my colleague Greta Wilt, who I showed earlier on a slide, made this map. This is not Nurses Health Study data. This is her data, but that is her residential address, her work address, and those red and black buffers that you can see there are the usual typical kind of close by, near home, and wider address. And you can see she does spend quite a lot of time in these buffers, but not all of her time. She definitely goes outside of here. She goes to work. She bops around there. This is Cambridge. This is Boston here. And the nice thing that we can get from this kind of very, very high resolution data is we have minute level collection, and we know if she was stationary, and we know if she was asleep, and we know if she was actively moving, or if she's going at a velocity that is way too fast for a human. Therefore, we say that's automated transport. She's probably in the car. So we know what she's doing. We know where she is over time on a very fine granular scale. And this can help us then create an activity space for Greta, who, if she is in a cohort, let's pretend Greta is a nurse, we can use that to measurement error correct the people even who we do not have that information for. So we have her estimate at the residential address, and then we have her real estimate, and we have everyone who's in this little sub-study. And then we can use that data to correct and say, usually we're off by this much, and people actually spend this much more time in green space when we actually have personal monitoring on them. So we can leverage that in a measurement error context. And so that's all been possible because of the GPS and the locational stuff. So all of this is satellite based. And we are not the only group getting very excited about this kind of work. So Meipo Kwan is in Hong Kong, and we had a symposium at the ISE in Taiwan this year that went very well. We had some great colleagues from the US and from Europe and across Asia. The best thing that I learned in this symposium is that Meipo Kwan is now back in Hong Kong. She was at UC Berkeley for a while. She's now come back to Hong Kong. She runs a geospatial group there. China is giving her enough money to put a satellite in space to do this research. So, like really going to blow us out of the water if we don't start catching up there. But very, very cool. It's all health related. I will say that we do have satellites. NASA has satellites. I'm American now. So NASA has satellites going up soon, I think, and Beate Ritz, who is in California, is informing them on how to best do that and collect information that would be relevant for air pollution. So you can also detect air pollution from satellites through the spectral signatures from the air. So she is helping guide them in how to best approach that, how to get enough exposure contrast and not just look at the high pollution areas, make sure you look at low and high so we have some kind of differences, and how that would best link with the health data sets that we have available. So we have NASA, but I hope that one of our environmental health departments has enough money to launch a satellite one day, but I don't see us soon. Okay, so on to modeling environmental interventions to reduce cancer outcome disparities. I actually changed some of these slides recently because Harry and I have gone back and we're doing a bit more work on this. But essentially, Harry is great at getting grants and he's pretty good, and won 2 awards at the same time. One was for prostate cancer research and one was for the Department of Defense. And they had kind of overlapping aims. But the Department of the Defense one was for postdoctoral level. And obviously he came here to you guys at Rutgers, so he couldn't take it with him. So I took it, which was great. And I've learned a lot by, we were in the same group, so I already had Francine as a mentor, Francine Layden, but Tim Rebeck also became my mentor, and I've learned a lot from having Harry as a mentor and Tim. And essentially, we're working on this cohort that Harry designed with Tim way before I came on the scene. And this cohort uses SEER cancer registries. We have 10 of them, and it follows men with prostate cancer from the year 2000 to the year 2018. It's rolling entry, so anyone who gets prostate cancer between 2000 and 2015 is included. What is different than most cancer registry studies in this case is that Harry and Tim have managed to convince these registries that we need residential addresses, and some of them are willing to give them as they are. Some of them require geomasking, so like a slight movement of the address in a random direction within a certain buffer so that we don't know exactly where they are, if it's rural, for instance, and we could identify someone. But essentially that's a big improvement for environmental health, because we're not having to do kind of county level analyses, which have all the problems of ecological studies. So we have all of these registries that are at the bottom. The focus is on Black African American men and white men to look at disparities. Most of the registries also just gave us everybody, and we have kind of Hispanic identifiers, so we could expand on that. And we are trying to add, before I head off to Oregon, a few more registries. So we're in contact with Georgia and Detroit and Oregon. So what do we have linked to these people already? And that's more than 800,000 residential addresses at time of diagnosis. We do not have residential histories right now, although we're also working on getting that from California and New Jersey. So we have air pollution linked already, Joel Schwartz's model. We have greenness, as I showed from the satellite earlier, the Landsat satellite. That's this Landsat. So that's 30-meter resolution. Data on how much photosynthetic vegetation there is in that 30-meter square. And then we've also added light at night, which has been linked to cancer in terms of, what is it called? Disruption. Circadian rhythm disruption. It also could be, very much anything linked to population density, because where there's light, there's high population usually. But there is some evidence. Trang Vo Pham, Fred Hutch is currently doing a personal monitoring study of light at night for cancer outcomes. Anyway, so we thought we'd have a look at that as well. And I won't go too much into the methods of aim 1, but what we've done so far is kind of looked at that in close proportional hazards models. We've made sure that we accounted for, we use an inverse probability weight for other causes of death, because many of these men do not die of prostate cancer. If they have slow-growing indolent tumors, then they will more likely die of something like cardiovascular disease. And in fact, the majority of those men, if they did die, they died of cardiovascular disease, not of prostate cancer. And we adjusted for what we had. So we have age. We had, we thought that for those environmental exposures, that diagnosis time period was very important, because the pollution levels in the year kind of 2000 are much worse than they are now. So we have a kind of cohort effect. Cancer stage at diagnosis, race, marital status, insurance type, neighborhood socioeconomic status, population density, and we debated whether to put state in there, because we do want to treat them as a whole cohort, but we just think that states have different healthcare systems, regulations, access. So we ended up putting state as a fixed effect in here. And it wasn't all linear. So we modeled using quintiles. So just to orient you, quintile 1 would be on the dotted line. It's not shown. But in the models, we have PM 2.5, which is the air pollution exposure, and then we have increasing quintiles. The results of PM 2.5 are a bit weird. We can talk about that afterwards. We will be adding NO2, which has much better kind of evidence so far for cancer outcomes. And then NDVI, what you can see is as you increase NDVI, so going quintile 2, 3, 4, 5, we get a decrease in mortality, especially cardiovascular disease, but we also see it for prostate cancer, which has been shown in HARI studies previously. And for light at night, we kind of see the inverse of that. We see higher light at night, and they are correlated in space, so we thought we'll mutually adjust. They remain intact. There are some light at night folks who are saying every light at night study, every green space study needs adjusting for light at night because they are very light at night focused. So this at least shows that both of those effects remain intact, even though they're, correlated and anyway, it at least shows that after adjusting for light at night, we still see the trend in greenness. And then we will be, well, actually, I've already done a bit of this, looking at effect modification by race and cancer stage, because obviously if you have an aggressive tumor, perhaps you're more susceptible to the environment around you or not, or we want to explore that. And then the original aim was racial disparities, so we also want to look if we could reduce overall mortality and reduce the racial disparities if we intervened on any of these environmental exposures. I'm also interested in having a look at a mixtures method, but just because I want to use the method. But it's probably going to show basically what I've just shown you in those graphs, but in a different way of presenting the results. We have had a student doing a hypothetical intervention. So I've looked at hypothetical interventions already, but I don't have time to show you. We've also had a student who has been doing the same thing, mentored by Hari and I, and Min-Yang Song, who is at Harvard. She has been using UK Biobank data, and then you have this deprivation index that I've shown you already. And we're seeing if the disparities in deprivation levels in the UK can be lessened with green space, with a green space intervention. So we've used this hypothetical intervention G-computation method to see if it would reduce. The results are coming out very soon, about to be submitted. We did see a reduction for total. We saw nothing for colorectal, which is Min-Yang's main focus, so not that great for his research. But we saw it was kind of mixed, but we saw a little bit of stuff potentially for prostate and breast, and the highest was actually for lung cancer, even though we're adjusting for air pollution in our models. So that was kind of interesting. That's got IFE Knack there because she did that work there. Okay, how am I doing on time? Not very long. So a very quick summary of what I'm planning to do when I get to OHSU PSU. So thinking about the exposome, I don't know if any of you have seen this figure before, but the exposome is essentially trying to bring together everything, everything since you were born that you were exposed to. And it came from, it drove the start of the UK Biobank project. It came from us doing the Human Genome Project and realizing that didn't completely explain disease. So then the exposome concept was written about by Chris Wilde. It was actually written about before that, but it didn't get picked up as much. So that drove kind of, okay, we should look at lifestyle, we should look at exposures like physical activity, chemicals, diet, and all of those things. And as well as that, the general external environment. So not just what we're putting into our bodies and that we have any control over and what we're doing, but also things that are, determined by society, including like traffic and education and, this social determinants of health is probably this bottom general external environment. And then that goes into your internal environment and, epigenetics, or you can look at all of your omics essentially goes into this framework. And then you have your health, your chronic disease outcome. So I would say my expertise is definitely more in this GIS, remote sensing, exposure, modeling kind of stuff. But this framework, I think, is excellent for communicating with people who are not in your field. So I've started to talk with cancer people. They don't talk the same as cardiovascular people, but we all kind of can understand this. And I think this is a really nice framework to start conversations with. This is an image just showing some outcomes, but this was from a microbiome paper, for example. I started to get very interested in the microbiome and the virome, and what externally living, non-living is kind of altering our omics and inner workings and resulting in disease. And I thought that was very interesting because microbiome, I didn't know so much about the microbiome, and I was like, but I know about those disease outcomes, and that's interesting that they're seeing such strong links there. And could that, for me, the interest is, can that be linked to the external microbial environment, and probably higher in green space, lower in cities, and is there anything there? So that for me leaves me with all the questions of still deciding what are the best data sets, how to measure them, do I want to use buffers, can I use any other technologies, do we just have residential location, are we measuring in space and time, can I also have longitudinal data? But I think the framework altogether allows me to focus on thinking of that and find colleagues who are working in those other areas, and we can all kind of brainstorm together about how to layer our expertise. So that's basically what I just said. So to harmonize data in space and time, it helps us to have that kind of framework. If someone's working on single cells, having that conversation about what I need to know and what they need to know, if there is any kind of overlap. I mean, single cells are a very extreme example of someone who's very far away, but if you're trying to think about cancer, then you really do want to do this kind of multi-level analysis where you're looking at all levels of the ways that you can look at this disease to try and work out what the causal mechanisms are. And what Harry and I often think when we're watching some of these talks is that, do they even collect information on social determinants of health? Do they have any idea where the cells came from, or who these people are, or where they're living, or what their living environment is? Especially when you're thinking about racial disparities, sometimes I think you can watch genetics lectures, and yes, of course, they have their race, and they can derive that from the genetics, but like what living environment, what are the epigenetic effects of the living environment that person has been in? Is that data collected at all? And I think this kind of framework can help us all to think about how to link into each other's work. And I just want to say that group specific rather than only aggregate analyses are definitely important, and where I am wanting to put effort into my research, because I think the masking of the effects in specific groups can often be very strong. So yeah, essentially carrying on looking at disparities and making sure we look at different groups and how important that is for policy, because policies don't fit everybody at the moment. And I am running out of time. So I want to thank all of the Masters and PhD students that I've worked with over this period, and some artwork there if you're interested that I did with the artists. And thank you all very much for listening.
Questions? Thank you so much. That was fantastic. Really, really fascinating. Questions? Yes.
Well, all right, I'll start. So this is kind of a tour de force, and trying to get my hands around the issue of confounding. The value of isolating confounders, it seems to me, is to be able to plan specific intervention that you could actually accomplish. But then if you go look at, if you sort of gradually come over to diversity as a key issue, and the kinds of intervention for dealing with diversity are very different, it seems to me, from those that deal with greenness. So how do you move forward from the data to application?
Yeah, I think a lot of that is looking at group specific kind of modeling. So then you can see the effect of those environmental factors in different groups. So you have a cohort and it's black and white like Harry's and mine. You can look at the different risks. And we also work on that hypothetical intervention modeling to kind of move forward from just saying, oh, look, there's a worse risk in Black people for air pollution. Like, okay, now what would happen if we reduced it? Would it close the gap at all? Would everybody's health improve? Would Black people's health improve more? So that hypothetical intervention is definitely getting at that. I think we're always hoping to eventually have enough evidence that it informs an RCT. There are RCTs going on greenness, which is impressive because it's very hard to control everything and work at that scale. There are kind of smaller scale ones on microbial diversity that I saw presented at IFE, which were really interesting. They had kind of sand pits with very diverse sand. It was from like a forest floor or just completely non-biologic innate sand. And they had kids playing that for a few weeks. And they did see some immunoregulation changes and took a lot of biomarkers from those kids. So there are ways to do kind of mini boiled down versions of seeing if any of those mechanisms really actually do anything internally to the body. But yeah, it is important to do group specific work to make sure that everybody is, we're not widening the gap by only improving the health of, say, white people. And yeah. Hope that answered you.
I was wondering if you can say a few things about your noise and CVD regulations, because you showed later CVD and the PM 2.5 and well, no, sources of noise are also sources of pollution because they are traffic, industrial activities. So how do you separate those? I mean, that's something that we have a big group, the Robert Wood Johnson, that does noise and CVD, and Professor. We have very long discussions on this, on how to separate the effects of the co-exposures to a number of other factors. So I was just wondering how you did it in this case.
In this case, where we were focusing on noise, we adjusted for air pollution in our models. And the air pollution models are much better than the noise model in the US. So we would expect that the air pollution model, we actually did expect this to be null. We expected that the air pollution model was going to be such better quality than the noise model that when we used it as a compounder, it kind of knocked the noise out. But we didn't. And I think that is because the US is very loud and does not have tight regulation on noise. And there's no reason to think the biological mechanism is any different between US people and European people. And we have a lot of evidence in Europe with some, nice noise modeling that the effect still remains even when you include air pollution in the as a compounder. But yeah, it is hard to pull them apart. I do think EVs offer an opportunity for us to pull them apart a bit more, because the air pollution actually goes down, but it's going to remain in a lot of ways from brake wear and tires.
When you talk about the pollution, I hope you don't mean just PM 2.5.
No.
You looked at reactive polluters like formaldehyde, across things that are related to internal combustion engines and traffic and things like that.
We did not look at formaldehyde. No.
Oh, I'm giving formaldehyde and O2. I mean, it's much better. It's correlated much more than say PM 2.5, which does not. So that's why it's not just air pollution in general. One related question, I don't want to spend too much time on this, but why did you pick the park service noise, and did you try to combine it with the Department of Transportation models of noise, which provide a much better resolution? Actually,
I agree.
Their mechanistic approach to calculate noise, I think it's much more solid than what
it totally is. And that is what I'm talking about when I say European models are better, is that they have residential address level of these mechanistic propagation models.
Yeah, but the US beauty model does also that. The park service model is just interpolated.
No, I know.
So I was just wondering why you picked that one.
The reason is that US DOT model started in 2016, and we wanted to go back all the way to 88. And on top of that, it's only modeled around arterial roads, and that's not where many of our nurses health study participants live. So then you would have to, on top of layer in 2 data sets, which is not like a terrible idea, but layer in 2 data sets, you would probably have to do some kind of estimate depending on how far from the road they are. But essentially we want to create something very similar. We have Knossos in Europe, and then the US DOT has its own model that's very similar for the propagation noise modeling. What I want to do is write a grant to try and do noise modeling in the US, and it is only just becoming really feasible to do something at that scale. Obviously Switzerland or the UK, it's an entire country, but it's as big as like a very small state. So like it is becoming computationally possible to do that now at residential address level. So anyway, if you know anyone at NIUHS, I will be trying to get funding to do that. Thank you.
Hi, and thanks for coming for a little quarter. I'm intrigued by your use of Fitbits to track your nurse participants because nurses can be incredibly busy, and for some who are in, say, surgical or procedure-heavy specialties, they may have to take off their Fitbit when they're working. So I'm wondering what sort of adjustment for that they do.
They did tend to keep their Fitbit on under their gloves if they were asked to. But yeah, they are a kind of weird cohort in that they work shift work. And we tried to track anything like that. And we asked them not to do shift work, not to leave the country, although one of them did go to Italy during one of her weeks, so she forgot. But they were supposed to kind of stay and do their normal life, and most of them did. Yes, some of them have very active jobs. So what with the geolocation side of things, what we could do is see if they were at the hospital that they work at. They give us all this information, so the nurses are very compliant, they have good follow-up, and then they told us where the hospital is. So we just essentially drew a buffer around the hospital. If they were, we called that work physical activity, which is very different from, leisure time physical activity, and doesn't always show the same benefits. So yeah, but that's a great question. We did have to dig into that after we got it back, and we were like, oh, they're very active, and it's mainly while they're at work, and unfortunately not when they're in parks, because they barely have any time to visit parks, it seems. But
yeah, I just want to say congratulations on your position in Oregon. I came from Oregon. It's a remarkable place.
do you also work like my husband for this day, because he is always telling everyone how remarkable it is?
Yeah, and especially with the collaboration with PSU and Oregon State, it's a really outstanding institution. Yeah, I have one question about the hazard ratios for green spaces, and could you kindly explain what you think is the result of the higher hazard ratio for the infectious respiratory for the green spaces? Is it because people hang out more often with each other?
On this graph, Minnesota, right? Yeah, that was a really weird result. Honestly, I don't have a good explanation.
That's quite interesting, because if it's about people gathering more often, then it's more like an open space, and more of the respiratory diseases happening indoors. This is very interesting.
As I said, this is pre-COVID, so infectious deaths are pretty much all pneumonia at this point. So pneumonia is a hospital-based. I don't know. I honestly have no good answer for you. So they essentially have public green space or other types of green space like parks near their residential address. Maybe they're suburban. I honestly have no idea. It's probably, if anyone has any ideas, I couldn't explain it.
I have an idea.
You have an idea?
In a meeting, one of the things that we've learned about in disaster response were the pollen storms or pollen related stuff. And I would imagine green space, where there's more green space, there's more pollen, where there's more pollen, there's more allergens, there's more lung inflammation, et cetera, et cetera. So that's what I saw when I saw that data right away. So, but we learned about all kinds of good stuff.
Yeah. We had no hypothesis whatsoever that it would be, we thought it was going to be null. So it was a surprise that we couldn't really explain, but that would definitely help explain. I feel like pollen would also be in the private residential gardens though, so then.
But not necessarily as much or whatever, depending on, what you're growing versus. In other words, most people don't grow ragweed at their house, right? And Great Big Park or something else might have a lot of it.
Yeah.
But the US also has an enormous pollen problem, which was it's man-made, because the decision was taken to basically just plant everywhere male trees, because they will not litter the roads and the parks with food or anything. So that's why we managed to create the pollen storms in the US, which are not common in other countries around the world.
I see we're at 1 o'clock, so I want to wrap up, but I know I saw your hand. Maybe you can grab Charlie before she runs off to her next appointment. But I wanted to thank everyone for coming. It was really a wonderful talk, and we wish you good luck with tours in Oregon.
Yes. Well, that sounds like it took them about 30 years to catch up.